Marty Goldman, just going to welcome you to the last Controversies in Cardiology of the academic year. Just to tell you, it really was, I think, an excellent year. I'll just give you a review of the people that we've had, and just in recognition of the great job that all of the fellows do. The monograph that they prepare, which is outside, many of them publish. I hope all of them get to publish it because it's really an excellent monograph on the topic. And Dan is going to present later today, his paper included. In September, we had Dr. Deepak Bhatt from Brigham and Women's, and Dr. Hung spoke about how low to go for blood pressure, lipids, and sugars. In October, Dr. Elazar Edelman from MIT, Harvard, Brigham, and Dr. Sheth spoke about bioabsorbable stents. In November, Dr. Hill was here from UT Southwestern, and Catherine Michaela spoke about personalized heart failure. In December, David Harrison from Vanderbilt, and David Narotsky was the fellow on microvascular coronary disease. Dr. Tomaselli in January from Johns Hopkins, and Robert Copeland Halpern on inherited arrhythmias. In February, Dr. Greenland from Northwestern, and Mohammed El-Kazaz spoke on asymptomatic patients with coronary disease, prevention and management. In March, Dr. Gold from South Carolina, Dr. McCullough spoke on arrhythmia device therapy and heart failure. Dr. Hall from Lillahei Institute in Minnesota, and Roman Shah on precision medicine. In May, Dr. Gerstein from Beth Israel Deaconess and Harvard, Dr. Shu spoke on diabetes and cardiovascular disease. And today we're really privileged to have Dr. McNally from Feinberg School of Medicine in Northwestern. And Daniel Moscanto is going to tell us about CAD and genetics. Dr. Fuster apologizes today. He's in the middle of flying to Europe, and we're going to have in his place our Director of Cardiovascular Disease and Genetics, Amy Kontorovic, but I'll introduce her in a moment. Dan.
Okay, thank you very much. Now that everyone's flash flood warnings have stopped, I'll talk a little bit about coronary disease and genetics. So despite continued advances in medical therapy and technology, cardiovascular disease remains the primary source of mortality throughout the world, representing at least 1/3 of all death caused by non-communicable disease. Coronary artery disease makes up the majority of cardiovascular disease and is responsible for over 350,000 annual deaths in the United States alone. Currently, medical therapies focus on mitigation and treatment of existing coronary disease, so-called tertiary prevention. However, as efforts are refocused on primary and primordial prevention of coronary disease before it becomes symptomatic, identification of those who stand to benefit from intensive efforts and therapies is becoming paramount. I hope over the next few minutes to introduce genetics as a realistic tool on the horizon of clinical practice that may help us solve this particular problem. Risk stratification for CAD is not a new concept. In 1948, the first patient was recruited into the Framingham Heart Study, and for the past 69 years, it has gone on to identify those at risk for CAD. Through the Framingham's risk score and similar risk calculators, we hope to identify those at risk for CAD and target our therapies towards those who benefit the most. There are some limitations to the predictive value of this score, however, specifically in those who are at the intermediate risk range for CAD. Supplemental tools such as the ankle brachial index, high sensitivity CRP, and calcium artery scoring have been developed to help risk stratify those patients further. However, the main drawback of these tools is that they require disease to already be present. As a result, these risk calculators are only aimed to predict 10-year risk and fail to identify those at lifelong risk of CAD. A doctor by the name of Carl Mueller described a hereditary condition where people were affected with xanthomas and had a proclivity of dying suddenly with symptoms of paralysis of the heart. He discovered familial hypercholesterolemia. Further studies would follow, and in 1994, the Swedish twin study examined the risk of CAD in twins and found a heritability of around 40 to 50%, a figure which has been confirmed in more modern trials. Investigations into these genes that contribute to CAD have been ongoing for decades. In the 1980s, a technique referred to as positional cloning involved binding DNA markers to individuals' genetic code, and through a process known as linkage disequilibrium, they could identify areas of genetic code thought to contribute to CAD. Further sequencing of these genes allowed for identification of the culprit gene. While useful in understanding lipid metabolism and perhaps in identifying those at risk for monogenic Mendelian drivers of CAD, this strategy left the genetic causes of CAD in the majority of the population unidentified. For most individuals with CAD, there are not one but several genes responsible for their proclivity towards coronary disease, each with a relatively small contribution. In 2005, a new technology referred to as DNA microarray or DNA chip became available. Like prior studies, it used polymorphisms in DNA, which we refer to as SNPs, to associate certain genes with particular diseases. However, it allowed the use of more than 1 million different DNA probes simultaneously. Development of this technology came at the same time as advances in computing power, which allowed processing of such data. Together, this led to what we refer to now as the Genome-Wide Association Study, or GWAS. This led to the birth of the Cardiogram Plus 4D trial, which included investigators from databases from several different countries, consisting of over 240,000 different patients. Analysis of this and subsequent databases led to the discovery of some 50 genes or so, which explain around 20% of the heritability of CAD. While this only represents a modest contribution to the causes of CAD, several authors have created genetic risk scores using the presence of these genes to calculate the risk of CAD in a particular individual. These scores have demonstrated ability to improve discrimination and reclassification of patients compared to traditional risk calculators alone. Furthermore, these scores excel at highlighting risk in younger individuals. Those patients younger than age 57 with a high GRS score have more than a 2-fold greater risk of coronary disease compared to those with a low score. For a score to be beneficial, however, it must not only identify patients at risk, but it must also show benefit in treating those patients. In a recent paper in the New England Journal, a genetic risk score was retrospectively applied to a database of 55,000 patients. It confirmed that those in the top quintile of genetic risk have a much higher risk than those in the lower quintile. However, with simple lifestyle improvements such as not smoking, moderate physical activity, and a healthy diet pattern, they were able to reduce that risk by as much as 46%. Further studies have looked at statin therapy in patients with elevated genetic risk score. Like lifestyle, those with high genetic risk also demonstrate significant benefit from the use of a statin, with a number needed to treat of 20 to 25 patients in high-risk groups compared to 57 to 66 patients in low-risk groups to prevent a single cardiovascular event. In conclusion, the increasing availability and decreasing cost of DNA microarrays make genetic screening a plausible tool in routine clinical practice. A DNA analysis could be performed once in a lifetime and assay for risk not only for CAD, but also other life-threatening diseases such as cancer and diabetes. While the addition of a GRS to traditional risk scores has thus far only modestly improved discrimination, further investigations continue to turn up new data that can improve current predictions. Importantly, the GRS score can be calculated at a young age where it has been proven to be most effective and allow for early intervention. It is easy to see intensification of efforts towards a healthy lifestyle at those who stand to benefit the most. Similarly, the use of a GRS risk score could be used to highlight those who would benefit from medical therapy, such as statins. The use of genetics in clinical medicine is just now becoming practical. As we continue to expand our database of causative factors relating to CAD and other diseases, it is hard to imagine a world where a DNA microanalysis does not become part of our routine preventative care. Thank you.
Let me just more formally introduce our visiting professor today. Though Dr. McNally was born in Chicago, she knew the best education was in New York, so she went to Barnard and then Albert Einstein College of Medicine for Masters of Science, PhD, and MD. However, she went back to her hometown in Chicago afterwards, and she had several positions, including after New York, she did her internship and residency at Brigham and Women's, fellowship in cardiovascular medicine, Brigham and Women, and then went to University of Chicago's Department of Medicine and Genetics. She was director of the Institute for Cardiovascular Research at the University of Chicago, professor, Departments of Medicine and Human Genetics, University of Chicago, and just relatively recently, in 2014, She became the Professor Elizabeth J. Ward, Chair, Department of Medicine, Director, Center for Genetic Medicine, Northwestern University Feinberg School of Medicine, and Professor, Department of Biochemistry and Genetics, Northwestern University. She's achieved spectacular accomplishments in her field of genetics. What she's highlighted as her major contributions: modeling rare variants for muscular dystrophy and cardiomyopathy, the first genetically engineered model of limb-girdle muscular dystrophy, the role of ferlin proteins in regulated membrane fusion events, the contribution of nuclear membrane to human phenotypes, as well as improving human genome analysis for cardiomyopathy, collaboration with the University of Chicago Computation Institute and Oregon National Laboratories to harness the power of supercomputing to improve the speed and accuracy of genome analysis. One of the things that Dr. Fuster always highlights is the direction someone takes early on. And what's amazing is her first paper, which was isolation and characterization of human myosin heavy chain genes, published in the Proceedings of the National Academy of Science, was actually while she was still at Barnard, with a senior author of a friend of Dr. Fuster's, Nadal Genard, actually. She has mentored over 50 postdoc fellows, currently 10. She has numerous NIH grants to continue her work, and she gave one of the most interesting, clinically relevant talks on genetics that I think I've heard at noontime. So it's really a great honor and pleasure to have Dr. McNally here, and I'd like to present you with this plaque in recognition of your visit. It says, the faculty and fellows of the Zena Michael Wiener Cardiovascular Institute and the Marie Jose and Henry Kravis Center for Cardiovascular Health express appreciation to Elizabeth McNally, MD, PhD, for outstanding teaching, wisdom, and expertise as the Ananda L. Sharma Visiting Professor and Simon Dack Memorial Lecturer, June 2017. Good. As I mentioned, Dr. Fuster is on his way to Europe, but we're in very capable hands. The moderator is Dr. Amy Kontorovic, who's the medical director of adult cardiovascular disease and genetics here at Mount Sinai, and we're really privileged to have her as the moderator. The panel will consist of Juan Badamu, Professor of Medicine, Director of Atherothrombosis Research Unit, Ron Doe, Assistant Professor in Genetics and Genomic Sciences, and member of the Charles Bronfman Institute for Personalized Medicine, the Center for Statistical Genetics, Raja Hajar, who's Director of the Cardiovascular Research Center, the author and Jane Ross Professor of Medicine, Robert Rosenson, Professor of Medicine, Director of Cardiometabolic Disorders, and most importantly and last is our fellow in cardiology, Dan Muscanto, who presented this afternoon. Panelists, please.
Okay, good afternoon. Dr. McNally, thank you very much for joining us. Thank you for your fantastic lecture this afternoon and for sharing your wisdom on this panel. Congratulations to Dr. Musicanto on an excellent monograph. So the title of today's Controversies asks whether genetics can answer the question of who. Who will get coronary artery disease? But arguably, the more important question, I think, is the how. How can we determine who's at risk? How can we intervene in the most effective way to reduce this risk? The cardiovascular community already has at its disposal a number of well-validated and robust biomarkers for predicting disease. We know LDL levels, HDL, triglyceride, blood pressure, weight, fasting glucose. These are the components essentially of our risk scores, and we actually already do a fairly good job of predicting or identifying the who. If genetics is going to advance the field, it will need to not just predict, but distinguish causality, because that's the path towards personalized medicine and drug discovery. Epidemiology, family, and other studies suggest that approximately 50% of the susceptibility to coronary disease is due to genetic risk, and breaking down the conventional risk factors into their individual components naturally lends us to a discussion of the genetic drivers of lipid metabolism. The syndrome of familial hyperlipidemia, which I'll now refer to as FH, was recognized more than 75 years ago, and it's been 30 years since loss of function mutations in the LDL receptor gene were linked with high plasma levels of LDL in patients with FH. Subsequent studies identified mutations in other genes, ApoB and PCSK9, as causal for familial hyperlipidemia and premature CAD. And this highlights one example of a rare monogenic disorder that underlies CAD risk in some individuals. The prevalence of FH is estimated at 1 in 500, although in some parts of the world that number is around 1 in 300. Dr. Musicanto, based on your experience and observations, how are we as a community doing at detecting FH?
Not particularly well. I mean, I think as a cardiology fellow, we typically see people with even just general hyperlipidemia after they've had sort of their first symptomatic event. So we see people with an MI or something like that, and that's typically how we diagnose people with hyperlipidemia. Even those with exceptionally high lipid levels, I think we treat them and most of the investigation stops there. I don't think much depth gets taken in terms of their family history, and I don't think much further investigation takes place in terms of whether or not they have FH.
I'm just going to pull up a slide here, which basically highlights how we're doing compared to our friends abroad. So even in small countries in Europe, where the total number of FH cases is probably about an order of magnitude lower than what we have here in the US, we're still falling far behind on our ability to diagnose. And I think this is due to the implementation of guidelines and standards in these countries, but it's quite impressive. If we look at the Netherlands, for example, is capturing about 3/4 of the predicted total number of FH cases. And we're really not even at the bottom of the iceberg, at the tip of the iceberg, we're at less than 1%. So along with a clinical assessment, genetic testing is incorporated into diagnostic guidelines here, but the usefulness of screening is uncertain. In several European nations, but not the US, cascade screening with genetics and lipid profile is recommended after identification of an index case in a family. Dr. McNally, what's your preferred approach for evaluating someone with suspected FH, and what role does genetic testing play in that?
I think it's a shifting landscape for that right now because, you know, if you had asked me a couple of years ago before PCSK9 inhibitors came around, most people would say, what's the value of doing genetic testing? Yes, we suspect it. Yes, we see it running in this family. We're just going to treat it anyway, so why do that? So that's where I think right now there's a number of, and to be clear, the limitation of why genetic testing doesn't get done is strictly just based on cost. There's a cost associated with it, and for many years, it has been cost prohibitive to do genetic testing. And so the answers to that aren't that the genetic testing isn't useful, it's that the cost needs to come down and then the utility needs to be shown. So it is valuable to identify who's at greater risk in the family and not just treat everybody, although you can use cholesterol to help follow that. But now with PCSK9 inhibitors, where the cost of the drug is significant and you really need to demonstrate that they need that drug, now we're seeing a number of third-party payers requiring that testing prior to authorizing use of the drug. And so I think we'll probably see an uptick in testing. We've seen it already at our institution, and I'm sure that will just continue. So now there is a clear reason to do it. But you know, it's also fair to just step back and say it's fairly artificial that the cost of genetic testing for FH is the price that it is. There's no reason it should cost that much money. And so if the cost of getting the test was like it should be, then more people would be tested. The reason why it's tested at high rates in countries with different healthcare systems than ours is the reason why.
Right.
Yeah.
And I should just say, I mean, not all of these diagnoses are being made on genetic grounds,
Yeah.
but I mean, the data from this table comes from clinical diagnoses as well. So I think even without the genetics, we're not doing that well.
I think that the difference between United States and Spain is because I am a member of the Spanish Society of Familial Hypercholesterolemia, is that if you have a genetic diagnosis, the government significantly helps you with the cost of the drugs. And then when you do is when you find somebody, you do the cascade analysis, but then because of the help of the government, the family is really very happy in collaborating. So it's not that there is, and the same thing happened in the Netherlands. So it's not that there is a highest frequency. What happened that if you don't look for it, you will not find anything. So I guess that this is the big difference with this table. And you had a good point. Money is a major issue, both in the diagnosis and what are you going to get if you allow yourselves to be diagnosed.
A number of years ago, the CDC actually listed testing and screening for lipid disorders as a tier 1 classification. So I think this is based on actually the high hit rates for testing in FH. Depending on the pretest probability, the likelihood of finding a genetic mutation can be upwards of 90%. In Europe, we should just mention that the cost per life gained for genetic cascade screening and intensive statin therapy for this disease is 3,000 to 4,000 euros. And this is really on par with the cost per life gained for conditions such as breast cancer with mammography screening. So I think we need to just shift the way we approach screening and testing. And I just wanted to highlight some of the proposed benefits or indications for genetic testing. These are just some factors that would suggest why it might be beneficial to do testing. One of the points here that's interesting, I think, is that what the studies in FH have shown is that for any given level of LDH, any given level of LDH cholesterol, the risk of coronary disease is higher among mutation carriers. Notably, for those in the highest LDL level bin, for those who have LDLs greater than 190, there's a 16-fold higher risk for mutation carriers. The other point to mention here is that the response to treatment is variable within individuals with FH. And statins are widely endorsed as the first-line therapy for hyperlipidemia regardless of genotype. Multiple studies have looked at this, and most of them have shown that LDL lowering by various statins is actually genotype dependent. Some have not found an association, and so in general, the exact mechanisms for genotype-dependent statin response are not clear. We know that the newer agents, such as PCSK9 inhibitors, yield quite dramatic LDL lowering effects. Dr. Rosenson, do you use genetic information to guide your treatment strategy with respect to specific statins? Are there any cases in which you would skip statins and go directly to monotherapy with PCSK9 inhibitors?
I want to get back to the point about diagnosis. I think that we all need to start examining the tendons in people with elevated LDL cholesterol, the dorsum of the hands and the Achilles tendons. And I'm actually quite surprised in some of my patients with very clear-cut heterozygous FH, compound heterozygotes, that nobody ever felt the tendons. And this is important because, as Amy mentioned, that CARA reported that if your LDL cholesterol is greater than or equal to 190, your risk for a cardiovascular event or coronary disease event is increased 6-fold. But if you have, LDL receptor defect or one of the pathways affecting LDLR, the risk is increased 22-fold. But it's also very important for cascade testing, which is identification of first-degree and often second-degree relatives. And I think that's our responsibility as a physician. So let's start with the exam. There are also tools that we can use to enhance our diagnosis, and the one that's the most validated based on genetic studies is the Dutch lipid clinics, and it was compared, versus Simon Broom and the MedPed score. I think the first author was Santos in the Lancet that reported that. We're very fortunate because we do a lot of clinical trials with PCSK9 inhibitors, and in patients with heterozygous FH, homozygous FH, DNA analysis is provided to us, but a lot of times it's insufficient. So we have, individuals that may have a suboptimal response to PCSK9 inhibitor. We can talk later about polygenic testing of other minor traits that affect the LDL cholesterol level because it's been quite insightful. And we've been very fortunate to have Rob Hegley run some of these analyses for us, gratis. But if I had the availability to, assess LDL traits in more individuals, if it was reasonable, if the insurance would cover, absolutely, I would, do this.
would it affect the way you approach treatment? I mean, do you see enough in the data to suggest that there should be genotype-directed therapy?
I think that the data is, I think, limited in terms of response to, therapy based on certain polymorphisms in HMG-R. This was work done out of the Brigham. I think it's more for prime prevention, I would treat earlier, but, let's think about this data from CARA, published in JACC, 6-fold versus 22-fold increase in risk. You're still going to treat somebody with a 6-fold increased risk who's got a very high LDL cholesterol level. I think that where the genetic data would help me is pushing for more aggressive treatment and perhaps transitioning past statin ezetimibe to, adding on a PCSK9 inhibitor, maybe a little bit earlier in the treatment, course. But, I often find that this information is very helpful for the unaffected younger adult who doesn't want to be treated because they feel that they're, young, to say, well, this is the genetic, profile of your sister or brother, and that you have a similar type of phenotype, and this is why you should be treated, and then go over the, the potential cardiovascular risk associated with, heterozygous FH. Thank you.
So a number of genes have also been associated with statin responsiveness, and those are proximal to the genes that we know that regulate LDL cholesterol levels, such as HMGCR, which you just mentioned, ApoE and LDL receptor genes, and also genes that encode the statin transporters. And several of the genome-wide association studies, which is a topic we'll delve into a little bit deeper in a bit, have shown associations of specific alleles with treatment response of different statins. So, for example, one study found that there was variable responsiveness or effectiveness of rosuvastatin, and that the effect was proportionate to the number of inherited alleles. And separately, other genetic modulators of statin toxicity have been identified, namely a variant in SLCO1B1, which confers an increased risk of myopathy with high doses of simvastatin. So, Dr. Hajar, given these findings, do you think that specific therapeutic decisions should be guided by genetics?
I think you have to look at what the value of each of these mutations contribute to our understanding first, and how they can benefit the patient in terms of the pharmacotherapy that we have right now. I think as an initial approach, these mutations are sort of guiding the treatment, in a sense that for each patient, you can sort of mitigate some of the adverse effects, especially with the myopathy, with HMG-CoA reductase inhibitors. But I think for a general population where you really need good penetrance, I don't think they're going to be extremely useful. Experimentally, yes, but I think for a general population where already we have such a low capture in terms of treatment, I don't see it in the very near future to be very effective.
Yeah, not enough there yet. Dr. Rosenson, I know you were involved with some of the GWAS that looked into statin toxicity. Anything else you want to add about that topic?
Well, it's really statin dependent. I mean, some of the traits like SLOB1, which is a problem with simvastatin, is not a problem with rosuvastatin. There's other ones with rosuvastatin that are not a problem with other agents. So, what we recommended in the National Lipid Association guidelines is you try a different statin with different pharmacokinetic properties. But yes, if we had the genetic data, then there would be certain people that may be harmed that we would avoid those bad decisions. Now, the other aspect, as I think about the genetics, we're well familiar that ezetimibe lowers LDL cholesterol by about 18%, but there's 1 to 2% of the people that have a 60% reduction. That's also under genetic control. Hegley reported that. And it would be nice to know who those people are before you, go from statin to PCSK9 and go through ezetimibe, first for somebody that's a hyperresponder that would get an expected response that's, in the ballpark of what one would achieve with a PCSK9 inhibitor at a much lower cost.
Yeah, I think that's an excellent point. Yeah. Dr. Bademan, do you think that the coronary geneticists should take home any lessons from what was learned about pharmacogenomics with P2Y12 inhibitors?
Yes and no. One of the things I wanted to say is that we already know that, especially for atherosclerosis, it's better to be proactive than reactive. So we already know that if a person has certain risk, we don't need to start spending money with molecular genetics. You know, you can already initiate the treatment very early on. And we already know that 10 milligrams of reduction very early on is more effective and has more efficacious than if you wait at the end for the patient to have a symptom and then you start giving atorvastatin 800. So one of the things is we have to be always proactive. What else I see that the genetics may be very important is in this, as Dr. Hayar says, it's the very few people that they do not respond to the normal drugs. This is a comment. Going back to your question is, again, one of the things is that I work a little bit in platelets and thrombosis, and we talk a lot about hyper and hyper responses to clopidogrel, but sometimes these hyper and hyper responses are lab findings, because not everybody that is hyper responder to Plavix is going to have the stent occluded, and vice versa. Some hyper responders, they also occlude the stents. One of the problems is, and this is what I like so much your presentation this morning, because you were very clearly separating monogenic diseases from polygenic diseases. And then when you talk about this genetic profile and you go together with atherosclerosis, we have to have very clear that in atherosclerosis there are three things that they play a role: the wall, the blood, and the platelets. And then it's going to be very difficult to be able to pinpoint what are the factors are going to be more important. In the moment that you go to monogenic diseases, then I am totally converted to your presentation and I learn a lot. This is what I say, that we have to differentiate totally in polygenic versus monogenic diseases. In monogenic diseases, early genetic study is highly recommended, especially if the parents have the symptoms. In the polygenic diseases, I am a little bit more skeptic about the benefits. What do you think?
I agree, and the problem comes in, and it's nice to separate monogenetic from polygenic, because again, we're really building the pathways of actually being able to get that testing and cascade testing and identify those family members. The hard part comes in that there's a gray zone of what is monogenic and what is polygenic. And just talking about FH is actually a good example of it, because we think of FH as clearly a monogenic disease. It's at a higher rate than what we really appreciate. There are also probably genetic variants that are kind of in between. They're not so pathogenic, but they also increase risk partially. So what are you going to call that person? They're partially at risk. Is it really a monogenic disease? So that it's nice to separate them, and we can do that effectively in some cases, but the issue is there is a gray zone, and understanding how modifiers change the expression of monogenic disease is really kind of a fancy way of saying polygenic. So.
But you see, if we put this in today's aspect, that our president is disagreeing with a lot of prevention, with a lot of money for drugs, how are you going to convince him to pay for genetic profiling if he's even against paying for anticonceptive pills or statins that they are clearly demonstrated that they do work? So, you know, this is the thing that is very nice when you talk about research, but then when you try to go to the reality, there is a big wall in the middle.
So Dr. McNally, before you turn off the microphone, I just sort of continuing on your train of thought, but shifting the question slightly, do you think that pharmacogenomics will have a place for improving the safety and efficacy of the statins and other classes of drugs that will come out?
Yeah, I think not so many years from now that will happen. And I think much of what we're talking about will probably, realistically, we're talking about cholesterol and even genetic screening that realistically will probably happen in children to understand what their lifetime risk is as we go forward. And one can easily envision that there is a standard set of pharmacogenomics that might be ethnicity specific or might not be ethnicity specific, but that you might have that small panel of pharmacogenomic genes determined at a relatively young age, so that you carry that information with you over your lifetime, and then that can inform those decisions about how to be treated. But we're a long ways, I agree. Certainly politically, we're a long ways from doing that. But is it, and I mean, here today, we're more talking about what's scientifically possible, and that is definitely scientifically possible.
But I don't want to be against genetic profile because I think it's the future. But as per today, I always remember one sentence that I think it was said by Jocelyn, and he said, "The genes are the powder that load the gun, but environment pulls the trigger." So my question is, what do you prefer? To have a healthy lifestyle from early on with a bad genetic charge, or good genetic charge, and then you tell the patient, okay, go for it because your family history is very good, and this guy starts eating pizza every day. You know, there is again, and I agree that the future is genetics, okay, and this is clear. But now the question is, what do we do today?
So I think CAD is actually a really interesting example, because while we, in cardiovascular disease as a whole, we are dealing with disorders that have a high heritable component. I mean, we talked about cardiomyopathies and heart failure earlier. I think that's a good case where there's a lot of genetic involvement in that. I think CAD is really a great example because you have a genetic predisposition, but you absolutely have an environmental influence, and probably even a stronger environmental influence than a lot of other cardiovascular diseases that have genetic influence. And so you ask me what I choose. Of course I choose good genetics and good lifestyle, but it's not the way the world works. But I will say, having run a cardiovascular genetics clinic for close to 2 decades now, most young people, now it's a biased sample because they're people who came and found me, but when you give them genetic information, it does, and they know they're at risk for diseases, often they take that information very well, and they do change their lifestyle to try to accommodate that and to try to improve their chances. I mean, I take care of people with inherited cardiomyopathies, but I say, hey, we also want to make sure your cholesterol is good and you don't smoke, and I probably have the lowest rate of smoking of anybody in my clinic because, again, these are pretty motivated individuals. So sometimes having the genetic information is especially motivating towards a good lifestyle, and so there is that possibility.
I totally agree. And I enjoy your point about doing the genetic testing early in life and sort of having that data on file for future events down the line. I think that's a very interesting perspective. So let's zoom out again now and return to the bigger picture of coronary disease. We know that the heritability of coronary disease is estimated at roughly 50%, and that FH accounts for only a small percentage of these heritable cases. Although the FH genes were discovered through family studies, efforts to identify other monogenic drivers of CAD have been disappointing. So here on this graph, you'll see I highlighted that when you look at the Mendelian mutations that account for FH, they fall sort of on the upper left quadrant, where these are rare mutations in terms of their frequency, and they have a large effect size. As we know, coronary disease is complex and common, and as Dr. Musicanto nicely summarized in his monograph, genome-wide association studies have been implemented to uncover other genetic drivers of CAD. For those who are unfamiliar, GWAS compare the frequency of DNA variants between disease cohorts and controls. And as Dan said, the first GWAS were published about 10 years ago, when 3 independent groups reported common variants at the 9p21 locus that were associated with a 30% increased risk of CAD per copy of the risk allele. Since then, about 60 distinct genetic loci for CAD have been found, and about half of these loci fall, not surprisingly, within well-established atherogenesis pathways. For example, lipoprotein a, which has long been held as an atherothrombotic biomarker, has been found through GWAS and Mendelian randomization analyses to be likely causative for CAD. On the other hand, GWAS variants explain less than 10% of variation for lipid traits. About half of variants identified through GWAS exist in this category all the way over here, where the pathway remains unknown. Dr. Doe, what is known about the loci that fall in this category? Are they revealing new mechanisms, or might they be potential novel targets for therapeutics?
So the answer is actually we don't know much about those genes in these pathways that remain that sort of remain uncertain. There have been specific hypotheses people are hypothesizing that these genes must not be associated with any sort of blood-based marker that we could measure, or else we would have found them already. So these are genes that are not involved in cardiometabolic traits, ones like lipids or blood pressure or diabetes. One hypothesis is that these are genes that are involved in vascular remodeling, so vascular inflammation. Maybe genes are involved in stable versus unstable plaques. But yeah, so the answer is not much is known about these genes yet. And so that's proponents of GWAS sort of like to highlight this and say, look, we've done GWAS, we found half of them, the mechanism is unknown. If we can do some functional studies and sort of pinpoint the molecular mechanisms of these unknown genes, then potentially we could find entirely new sort of biological pathways for CAD in which we currently do not know that exist outside of sort of established cardiometabolic traits.
Dr. Badaman, with these discoveries, do you think we've solved the genetic basis of coronary disease? Do you think that this panel of variants that have been identified or genes that have been identified is the end of the story, or do you think there's going to be more to it?
No. For instance, what we know that when you use one of the global criteria score, there is always some like a gray area, especially in the patient that you qualify as a middle risk. You don't know what to do, whether you should really be proactive and put, for instance, high potency statins, or you don't care. So we know that in this population, if you do a further analysis, you are able to re-stratify a significant number of these middle risk. I think that here is where the genetics could be interesting and it will be cost effective. But to run to everybody, again, in an ideal situation is fine, but today when there is people that cannot afford statins, or the major problem is that the people do not take medicines. So, but the question is, in this population that you don't know what to do, then I think that the further genetic analysis will tell you whether you have to keep one notch the preventive or whether you can go down. And obviously, once this is small workout, it will be better than calcium score, for instance, because this does not involve radiation. And then you can get a significant more information. When I say more, it means a higher number of genes. But to do to everybody, I think that we are very far away. But again, don't misunderstand me. I think that this is the future and we need to continue. The question is we have to differentiate between what is going to be research and what is clinical practice.
Yeah, please.
Yeah, so one of the issues that we have to discuss is that the biomarkers that we're using in these studies also have their limitations, right? HDL cholesterol was, it's considered, the cargo in HDL, but not functionally related to the atheroprotective properties. But, where the studies have looked at HDL particle concentration, which have been a very robust predictor of events, and then recently in the Jupiter trial, more important than macrophage cholesterol efflux in predicting events in that trial. So,
we're going to talk about that also.
Oh.
Yeah, no, that's okay, please.
Well, so I'm just saying that some of the conclusions we make are based on what's readily available in large studies, but it's an incomplete picture, based on, a particular biomarker. And it doesn't mean that we're not going to learn more in the future about HDL by, expanding our, analysis of particle number, HDL proteome, lipidome, more specific, structure function relationships.
So I think what everyone is really touching on is that we haven't really reached the full understanding of the heritable risk for CAD. We only covered about 20%, which is what Dr. Musicanto has already mentioned. And one explanation, which was proposed by Dr. Badaman, is that there's likely gene-environment interactions that require a modifier for the gene to kind of be unmasked and have its effect. Dr. Musicanto, what else explains the limits of the GWAS studies to date?
Sure. So there's a couple sort of well-accepted limitations to the GWAS studies. I think the first, and something that Dr. McNally already touched on during her lunch conference, is that the GWAS study looks at common variations that are present in a large portion of the population. So individuals that have a rare variation, even if it's not having a major contributor, may not be picked up by these GWAS studies. I think there may also be variations that aren't analyzed by this, things like epigenomics, or there can be repeat sequences that aren't picked up by the GWAS studies, may also have a contribution. And finally, in order to try and prevent picking up false positive genes, these GWAS studies have a sort of a very high, sort of rigorous p-value, I think p times 10 to the negative eighth, in order to define a gene as significant. And so if there are genes with a relatively smaller impact on a particular phenotype, CAD, in this situation, it may not reach statistical significance easily, even with our databases of 240,000 plus patients.
Dr. Hajar, most of the signals actually are outside of the protein coding region. Are you surprised by this?
No, not necessarily. I mean, I think the disappointment in the GWAS analysis of, I mean, you pointed them all out, but I think the next generations for RNA sequencing is giving us much more data and some surprising data. So StarNet, which was a study that was run in Sweden and with Johan, who's here at Sina, was published last year, and they took cabbage patients and looked at different tissues during the procedure, and the RNA sequencing showed, very surprisingly, that fat tissue was or fat signaling was one of the most important predictor of coronary disease in this population. Okay, it's a cabbage population, very specific. So I think we have a lot more to learn, beyond GWAS. And RNA sequencing, post-translational modifications of proteins are going to be areas that are going to be important. I mean, the question is, on a day-to-day basis for specific patients, what is it that you would do that would be different? We went from talking about genetic testing for trying to understand the side effects, which can probably 90% or 80% can be solved by changing the drug without having to do the testing, all the way to now we're in a situation or a reality where if you are dubbed as having a monogenic disease, you may lose your insurance, right? I mean, familial hypercholesterolemia already puts a stigma on your insurance. And then if you have a genetic disease, monogenic disease, I mean, I'm more familiar with the cardiomyopathies where that's the case, where the premiums really shoot up. So what would happen if you have a genetic signature in your medical profile? What's that gonna do to your insurance? So I mean, from a molecular aspects, I think we have a lot more to learn, and we will with the new technology. But I think we just have to be practical vis-a-vis the patient also.
Dr. McNally, do you want to?
Let me be positive, okay? So at the same time with GWAS, we learned something that is called the PCSK9 inhibitors, that in 10 years, it went from a pure observation to be a drug. So we can learn certain things, but the problem is, Roger said, how are you going to deal with the stigma? In one side, it's good that you know, because then this tells you that you have to be more careful. But on the other side is, how is the society going to use this information?
Please.
I just wanted to add as well, Amy mentioned that most of the GWAS variants are non-coding, and so it's actually still quite difficult to link the non-coding variants to the expression of a gene. Like both computationally and experimentally, it's hard to identify the actual gene involved.
Well, I mean, unless you do RNA sequencing where you start seeing these long non-coding sequences that seem to do something, right? I mean, this is where
RNA sequencing would help, actually. Yeah.
But often the gene is very, very far away from where the SNP is, and so it requires different methodology to get at it. RNA-seq being one methodology that can help, but there's other methodologies that can help it. But yeah, still to this day, many GWAS signals, exactly how they exert their effect is not known.
So let's just go back to HDL. It was brought up by a few people, Dr. Rosenson, Dr. Bateman. So it seems like we've established causality for LDL and triglycerides, which we'll talk about a little bit more. Dr. Doe's been very involved in those studies. But we haven't established causality with HDL, and I think that was surprising to some because of the longstanding association between, inverse relationship between HDL levels and CAD risk. So Dr. Madiman, what do you make about the paucity of genetic data for supporting HDL causality?
I am very biased when you touch the HDL because I am a fan of the HDL. And then one of the questions that we have learned is that the HDL seems to be more complex than we thought at the beginning. Because now we say that the HDLs, they do not work because the more recent pharmacological agents, they do not work. But my question is, why they do not work? Is it because the HDL does not work, or is it because those drugs, they raise a dysfunctional HDL? And I am very carefully using dysfunctional because you already mentioned one of the papers done by Kera that where the HDL plasma levels is not so important, but when you look at the capability of the HDL of removing the cholesterol from macrophages, this is one of the best markers. So, and the question goes that biology already tells us something like that, because we had for a while a recombinant APOI1 that it was called APOI1 Milano that didn't make it to the market because it cost $25,000 an injection. But the people that generated the APOI1 Milano had an HDL of 0. So, and they didn't have cardiovascular diseases. So this is what we have first. We have to understand what is the meaning of this protein. And when you know what the protein does, then you try to understand. Because from my point of view, even though HDL does a lot of things, for me, the major role is to remove the excess of cholesterol from the extrahepatic tissues and carry it back to the liver for metabolization. And then sometimes I always make the same joke. What do you prefer? Do you prefer an 18-wheeler truck already full of cholesterol, or you prefer a pickup that is empty? I prefer the pickup empty. And what the CETP inhibitors they did is they generated this HDL fluffy, rich of cholesterol, that this is what they call it dysfunctional. I mean, because biology already told us. And the other thing is, Bob already mentioned the particle numbers is more important than the plasma level. So here we have that we try to explain with genetics something that we don't understand the physiology of the HDL. And then if we don't know how this protein works, we cannot explain it with genetics. So this is my point of view. But
Dr.
again,
Bateman's not a fan. What do you
but
think?
again, it's a very biased opinion for two reasons. Because I like the HDL and because I am from Spain with Mediterranean diet.
Right. So are we done, Dr. Rosenson, with HDL? I mean, the genetics
No.
closed the book?
No, not at all. So HDL cholesterol is a robust marker of reduced cardiovascular risk, but HDL cholesterol is inversely related to triglyceride-rich lipoproteins, which are clearly in the genetic pathway. Some very strong Mendelian randomization, studies support that. And also, a lot of the HDL cholesterol is explained by lifestyle changes. And we have a paper that we presented from the REGARD study that, showed that, diet, exercise, cigarette smoking, alcohol, et cetera, explains a lot of the risk associated with HDL cholesterol and cardiovascular disease. We already talked about HDL particles, but, we shouldn't forget about the data from Heineke and the JCI that, HDL carries a rich proteome. Initially, he identified 47 different proteins. Now it's 90 have been identified, and many of them are involved in immunomodulation, anti-inflammatory effects. It may be that HDL is providing protection there, but until we have the assays where we can do high throughput and get those signatures identified and then tested in a GWAS Mendelian randomization, we're just not going to be able to solve those problems. I just want to make one point where I was really disappointed. So I actually had the great fortune of running these international summits on HDL biology, and we went through nomenclature, efflux, functional HDL, dysfunctional HDL, and evacetrapib seemed to have all the right components. In terms of HDL cholesterol went up, HDL particle concentration, macrophage cholesterol efflux, so we thought this was going to work, and then of course it failed. And now, is this the CTP pathway, or is it the fact that our biomarkers that are, shown to be associated with reduced cardiovascular events in observational studies are just not valid when you're dealing with a post-ACS population who's treated with high-intensity statins? And then you look at the Jupiter trial, the efflux assay was less important in people on Rasuva 20, than it was in the placebo group. So again, I think everything is context, driven. And I don't think that we can go back to Framingham and some of these older studies and say, well, boy, HDL cholesterol is the best thing because it's a different era and it doesn't take into account a lot of the newer therapies or the current contemporary therapies that we're using right now in the ongoing studies.
So, Dr. Doe, I want to ask you, because you were involved in several of the GWAS that brought to light the role of triglycerides. I think, clinically there was some debate about whether triglyceride lowering actually played a causal role in protecting against CAD. But I think the GWAS have been very convincing in showing that there is a protective mechanism there. But why do you think the triglyceride lowering therapies haven't come through as the save-all here?
So both GWAS studies and also sort of rare variant studies have shown that altered plasma triglyceride levels have also shown to affect risk for coronary artery disease. As Dr. Controversies noted, that's sort of there's a discrepancy in the clinical trial literature where therapeutics that target plasma triglycerides have not had an effect on CAD. And I think there's a couple things. One is the dose response isn't strong enough for these therapeutics. And then a second thing is when you're looking at genetics, this is inherited genetic variation that represents like sort of a lifelong risk to disease. When you take a therapeutic, that's sort of like a acute thing that you only take over a very short period of time. And so a lifelong risk obviously does not equate to something that you just take over like a few weeks. So those are sort of...
Do you think maybe we've also not found the right mechanism for lowering triglycerides?
That's also possible because plasma triglycerides, we don't actually think it's triglycerides. We think it's triglyceride-rich lipoproteins that are the atherogenic component. And so, but we're just using plasma triglycerides because that's the easiest thing to measure in the blood right now.
Thank you. It's good to have your input since you were the first author on the papers.
So just one other, just brief comment is the mechanism, the most established one is ApoC3. Yeah. And not all these triglyceride-lowering drugs reduce ApoC3. So in order to test this hypothesis more accurately, you have to look at high triglycerides, associated with ApoC3 containing remnant-like lipoproteins. So again, this gets into the limitation of a biomarker, e.g. triglyceride, because there's much more to the story. And the more that we can refine the biomarker that's used in these studies, the more likely we're going to get reliable and reproducible answers.
So we know there's a massive data now from the GWAS, and they've uncovered about 100 different loci involved with lipid metabolism or coronary disease. Dr. Hajar, what type of studies do you think are necessary to obtain the greatest yield from this data that we have?
Again, I mean, I'm biased against GWAS. I think some of the more sort of precise ways of looking at this is usually taking these specific patients with these variations and doing IPS work and trying to understand how vascular cells and endothelial cells within those patient population behave differently or not. And that would be sort of the first sort of specific way of looking at it from a patient's perspective. But then you have to sort of kind of connect the dots from the actual variation to phenotype change, if there is any. I would say that probably 99% you would not see any phenotype change in any of the usual physiological studies that we would do. But again, this would be my. I mean, I think we're beyond GWAS now. I think we're in the post-GWAS era. And yes, we can learn certain things from GWAS, but I think the analysis in the future will go beyond GWAS, and hopefully will give us a more precise mechanistic information.
I think the GWAS give us the targets, but we have a long way ahead. I mean, Dr. McNally, what do you think?
I agree that I think the real value from a lot of the GWAS studies is really getting at the mechanism of how they work. So are they simply a marker that's linked to what the actual action is, or are they themselves the driver? And if so, how are they acting? What genes are they affecting? Because once you get at those pathways, then that's how you get at new drug targets. But I think the point that was just made, which is to also really understand very carefully what it is that's linking to those GWAS hits, just measuring triglycerides, or how are you looking at LDL or HDL, really being able to define your biomarker that's being linked to the GWAS is equally critical. So it does require more investigation, both to get at the mechanism and to make sure we're linking it to the right trait.
So I just put up this slide to show what type of complex pathways or algorithms in research would be necessary for each individual locus to try to understand, just as Dr. McNally is saying, what the connection is with the biomarkers and how it manifests perhaps as disease. So we have a load of SNPs that have been uncovered through GWAS, but each individual SNP has a very modest risk associated with it. And so the incorporation of one SNP into a risk score is probably not going to augment the data in a meaningful way. And for this reason, people have proposed the use of combined polygenic risk scores to try to better capture an individual's risk. So Dr. Musicanto, can you explain to us a little bit what is a genetic risk score?
Sure. So I think you sort of touched on it already, but the idea is that these are very common things that are common alleles and mutations that convey a small additive risk to causing coronary artery disease. And so if we sampled any single individual, they'd probably have some of these. And so in order to use genetics to help risk stratify, we have to come up with a way of sort of standardizing genetic risk. And so, looking at the New England Journal paper, for example, they took 50 different alleles and basically had a cumulative risk score for each patient, considering the relative risk of each allele and the number of alleles present in each individual. And then they standardized that and took the top quintile and the bottom quintile and compared them in terms of genetic risk.
So I think the benefit of these risk scores probably falls in helping to clarify ambiguous risk. Our current tools work well in the high and perhaps the low risk groups, but as Dr. Musicanto presented in his introduction, but in the intermediate group, it's not as clear. Dr. Badaman, are there signals for genetic risk scores being more informative in certain groups? Now I'm thinking beyond just intermediate, low, and high risk, but what about men versus women or certain demographic groups, ages, anything that you're aware of?
I already said what we can get, but one of the problems that, what I apply to the middle risk also applies to men and women. We know that we are not biologically equal, and the incidence, and this is one of the other problems, that sometimes a lot of genetic studies are doing in populations or samples taken from clinical trials that are already stored. And sometimes women, they have a very reduced presentation. And then all the studies that we are doing here, when you talk about genetics, you extrapolate to men and women, but probably, and you will know better than me, the number of females is significantly reduced. And then we are extrapolating there. I think that we are at the beginnings of what we are learning with the genetics. And you have to take into account, as Roger said, the first step is to take the patients with the disease, but again, all data that you get from there are significantly biased, because you are getting for a predisposed population. And then you have to start moving to the general, and then you have to start to move. So I guess that, yes, it's the future, but we don't know still what to do. And the other thing that I wanted to ask Dr. McNally, if you allow me, is don't you think that the technology is going much faster than the statistics? That we are getting data and data and data and data, and we don't know which algorithm to use?
So a couple of points. One, I agree that the samples that are often used for a lot of these big studies may not be so representative of the population as a whole. So one is sex bias, but the other is the ethnic makeup of a lot of the groups. So much of our databases, as they've been built, have been so biased towards the Caucasian population, which, genetically speaking, is just a highly interrelated group of individuals. And so as we expand and have better, more diverse populations, we learn that diseases look different in different backgrounds, if you will. And so I think we're in the forefront of seeing that right now as those databases are being built. The question about whether the statistical methods are able to keep up with how quickly we're generating data, I think that's exactly right. Sequencing has now become really cheap, and you can generate gobs and gobs and gobs of sequence. And the real limitation right now is analyzing all of this data that we have. And I say that not only about the statistical methods, which new statistical methods are being developed all of the time because the nature of what this data looks like requires new statistical methods. Multiple testing kills so many of our results, and yet at the same time, I think you were bringing up with p-values, we know there's still a lot of data there that we've passed over because of multiple testing. So we know that that's not a perfect method for when you're analyzing 4 billion, data points. But at the same time, computationally, we also have a challenge, which is being able to, how do we analyze this data? So yes, we can generate all the sequence, but we still need better computer approaches to looking at the data, and that's everything from aligning it and calling it, and then the statistical methods to begin to look at what all this data looks like. We haven't developed them yet.
And the other thing is, how do you separate the redundance in the mechanism? Because I just did, I am more in translational, and I did a preclinical model. We use a multiplex, and we got a lot of information. And then, for instance, certain inflammatory markers are upregulated, and then the P-selectin, all these adhesive proteins that are released by the endothelium that are also upregulated. And then my question is because the inflammatory status, or is because the release of the protein, and then, is the typical story that you say inflammation triggers thrombosis, but thrombosis triggers inflammation. And when you do these studies, you know that everything is upregulated, but you don't know what is the cause of the consequence. And I guess that the problem is that we see something, but we don't know how to evaluate it, and it's a problem with the algorithms. And this may be the confusion that we are having, as you pointed this morning very well, that some of the mutations, if you go to the database, the exact and the other, in one says that is very risky, the other is not risky, and if you go to 23andMe, they say, do not worry. All this confusion probably is because we don't know how to really work with the magnitude of data that we are generating.
So including Dr. Fuster's recent publication co-authored by Dr. Babber and Dr. Moran here, the implementation of polygenic risk scores seems to be based on GWAS or conglomerate meta-analysis from studies that included mostly European individuals. Dr. McNally, you already mentioned this, but do you have concerns about implementation of a score more broadly to other racial and ethnic groups?
I do, just for the reasons I said. When you look at the genetic diversity among Caucasian individuals as a group, it's a really narrow group. And while it's been useful for a lot of studies and probably allowed us to do some studies on smaller size cohorts, it's a wide open question whether that will extrapolate to other groups. We simply don't have that data yet for many things. And so as we build our biobanks, it's really critical to build ethnically diverse biobanks. And I know here at Mount Sinai, that's, this has been a really important thing. You have a very diverse population in New York City. We have a very diverse population in Chicago. So we're involved in trying to make a diverse biobank as well. But it's a challenge because, people don't always want to participate, and yet we have to work really hard to have very inclusive data banks.
Yeah, there's a historical issue there too, of course. Dr. Hajar, do you think that there's a role for studying microRNAs or anything coming to the forefront in that field to try to enlighten the issue?
Yeah, I mean, I think you know, so the explosion of information is already here, and we have to sort of narrow the phenotype more and more on these patients, try to get really specific patient population. They may not be totally representative of the whole coronary disease spectrum, but because the explosion of all this information, the phenotype at least will have to be restricted so that we can understand better and analyze better the information that we're getting from RNA sequences, all the way from, microRNA to long non-coding RNA. It's going to be a lot of information, so the phenotype has to be very specific. And you may, we're going to get back to monogenic diseases where you're going to have very specific phenotype representing coronary disease or one aspect of coronary disease, linking different RNA expression, and that's going to be very useful, I think.
What would that look like, a specific phenotype, because it's so complex?
I mean, you may get, let's say, young patients with specific calcifications of the coronary arteries that are, now are being seen because of MRI and CT scans that have a very different signature from someone who has very premature coronary disease with obstructive coronary disease. So, I mean, these are the types of patient populations that are going to be useful. I mean, I think in cardiomyopathies, that's very pronounced because we have specific mutations that can cause cardiomyopathies, or, one are very different than the others, but you have a specific phenotype. So, I mean, I think that's part of the one brave idea from the American Heart Association, really, to connect the phenotype with the genome. And I think we're going to hear about it tomorrow, since Callum is coming tomorrow, right, to talk about this. So,
look forward to it. Dr. Joe, can you tell us whether there's anything on the forefront with rare variant association studies? So we talked about GWAS, which rely on common variants in the population, but I know you've done some work in rare variant association studies. Should we expect that will help, add more and enrich this topic?
So sort of that's sort of like the next step in terms of human genetics. GWAS studies have found hundreds of loci for coronary artery disease, and now people are moving towards sequencing studies. What we found so far is there actually isn't much in terms of finding rare mutations in genes that contribute risk to coronary artery disease. Whether or not that's because there isn't much there in terms of actual rare variants contributing to the disease is unknown, or whether it's a matter of sample size. So right now we're at the scale of tens of thousands, and people have done like power analyses and simulations based on the observed effect sizes seen today, and they show that the current genetic studies, even at that scale, is underpowered. So it kind of remains to be seen whether rare variants contribute to coronary disease.
So I just want to shift topics to the last topic of the discussion, which is about how to implement this information toward therapy. And I guess I have a question for both Dr. McNally and Dr. Hajar. Whether you think there's anything on the horizon with gene therapy, with gene editing. And I guess, Dr. McNally, I'll ask you first.
So, I mean, we've seen a number of remarkable things happen in genetics over the last probably couple of decades. I mean, the huge big increase was just being able to do PCR and get a hold of things. But then you fast forward to the idea of next generation sequencing, to being able to do iPS cells, and then now being able to do CRISPR. So you realize that setup now makes it possible to, even from an individual patient, come up with identifying a mutation that's responsible for their disease, building a cell-based model, and then being able to test, at least in that model, and show that you can do some correction. And so now we're faced with how do we think about how do we get this into patients? So I'll let Roger talk about it a little bit more. I mean, our limitations about actually thinking about doing gene editing in patients are several-fold, putting the ethical issues over here, because we could talk about that for several days. We still are limited by having to deliver using a viral system, because we don't have another way of doing that at this point. We still have to probably improve efficiency a little bit more and fidelity of the process, because we can't have off-target effects. So I think we'll still see some optimization in terms of the machinery that does gene editing. We will see that coming in the near term. And then we still have to think about the delivery mechanisms of how to get it in and have it be efficient. But that said, there's been enormous progress just in 3 to 4 years' time. So while it's easy to say, oh yeah, in 10 or 20 years, which is still a very short period of time that might be happening, it could come, and I actually expect it to come sooner than that in humans.
Dr. Jar?
I mean, I think the basis of gene therapy or gene editing really is that you have a specific mutation, a specific gene that's causing trouble and or dysfunctional that you're correcting somehow by either loss of function or gain of function method through your gene therapy or gene editing. And I think that's going to be really important in terms of coronary disease, that to establish these relationships between a causative variant or mutation and the disease state, because as Beth very nicely said, I mean, there are ways of editing the genes, of doing gene therapy, but there has to be certain correlations along with the whole list, as Beth mentioned, of delivery, specificity, safety, off-target effects of these procedures. But I think, at least in cardiomyopathies, we're getting closer. In coronary disease, I'm not exactly sure.
Yeah, Dr. Badamon and Dr. Rosenson, same question.
I am surprised that there is a concept that has not been mentioned, either this morning or this afternoon, that is epigenetics. Because we talk about gene, but we are forgetting that, for instance, the Japanese, they have strokes more than MIs when they live in Japan. And 15 years after they move to San Francisco, they have the same MIs that the Americans, and less strokes than in Japan. And the genetic chart is the same. So I guess that there is something that we have to factor in here, and then we will learn a lot. One is that one gene may behave in a different way when it's exposed to different environment. This is what they say, it takes two to tango. So that if we don't know what to do with the genes, now when you put the epigenetics, it's still more complex. But what do you think about this new term, the epigenetics?
No doubt, epigenetics is really one of the clear ways we now can understand how environment influences the expressivity of a lot of these mutations. That is a very clear mechanism that, the details, the specific cell type, the specific genes, how it's being shut down or turned on, that's all to be worked out. I think when we talk about gene editing, we're really, again, stepping back to thinking about a rare Mendelian disorder where we clearly know that mutation causes what is a very bad disease in that patient. And so those will be the targets for gene editing, where to be very clear, the genetic effect is the strongest effect, and epigenetics can't even do very much about it.
Well, there are some examples in the lipid field where people are getting transfected for certain genes like the LPL. But I don't know that that's being marketed anymore. It was $5 million per treatment, and then it in Germany, the LPL. 1 million. Okay.
It was 1 million.
All right. And then right now, I mean, we were approached by Dan Rader's group, for some of our homozygous FH patients to introduce LDLR, but we're not going to participate in those studies because we've got some other ones with angiopoietin-like 3 protein that seem a little bit more interesting and to us. But there are those opportunities for some of the real extremes.
Thank you all for your comments. So it's been a very exciting and provocative dialogue. I know we're all looking forward to what's next. Did you want to add something, Dr. And I want to thank Dr. McNally again for bringing your wisdom and expertise today, both this afternoon and this evening for the panel. And thank you to all the panelists for participating. Just to end the session, I just want to ask each of you one by one, where's the revolution headed, and how do you see cardiologists implementing these genomic tools into their practice 5 years from now? Dr. Doe, I know you're not a clinician, but perhaps you can share your vision with us, and you can start.
I think it's actually just what this whole panel is about, which is like genetic risk prediction and trying to make it so that's clinically useful in both the clinic and at home. So there's a number of sort of genetic testing companies like 23andMe and Ancestry, and people are signing up to these just out of curiosity. And I think the next step for those type of tools is to actually demonstrate clinical impact.
Yeah, I mean, I think a lot of what we talked about, a lot of what I spoke about in the monograph is the use of a genetic risk score and things like further risk stratification in people who would otherwise be at intermediate risk. In other words, to try and supplant, say, a coronary artery calcium score. And I think while there's certainly a lot to be explained, left to be explained, as we've mentioned already, I think we are already seeing studies that have shown benefit in discrimination and risk stratifying patients. And I think as prices come down, it will become a reasonable strategy for us to help determine whether or not we really need to start the statin in this patient.
I think cardiologists are going to have to get a lot more comfortable with genetics, because I think you can teach genetics to cardiologists more likely than you can teach cardiology to geneticists.
I think the field is evolving in very important ways, and our use of genetics has not just been from the clinical trials, but trying to understand some of the extreme phenotypes of somebody, let's say with FH, worse than a heterozygote, not as bad as a homozygote, who ends up having a major trait, a monogenic trait, but then a polygenic risk score that's over the 90th percentile. And then when we're asking why are they not responding to statin, ezetimibe, and PCSK9 inhibitor as much as we would have expected, well, they have a big genetic stack against them, and that really helps with the understanding. Similarly, we just published a case of somebody with a chylomicronemia syndrome. So triglyceride levels, maybe 4,000 or 5,000 symptoms. He had tendon xanthoma, so you knew he had FH. And Rob Hegley, did the triglyceride testing for us, and he had something like 26 out of 30 or 20 out of 30 of the polygenic scores for triglycerides. So, our models of thinking it's pure hypercholesterolemia don't fit. And sometimes genetics provides an important clue, and it's helped in the, counseling of that patient who, really needs to stick with the diet and not have that six-pack of beer when he goes to the baseball game, to say, you've got to work harder than the other person because you've got so many genetic traits that are working against you. So I'm finding it very useful. And you brought up the, critical issue about drug safety. I mean, that's important. Boy, I'd love to have that information before I made the decision, before I found somebody got into trouble. Yeah, I'll switch them, but, not everybody makes it through without damage, on their first statin, where they may have rhabdo and acute renal failure.
And just to harp on that, in follow-up to what Dr. McNally said about motivation, patients who've had an adverse effect to one medication may be reluctant to comply and adhere to another. Dr. Badalone.
For me, it's easy. Once the cardiologist learns and understands the power and the information that all these techniques, whether it's gene or microRNA profile, when you can digest this information, then the application will be immediate. The problem is that we have to learn. And I think that this is why it's so important, the lecture like this morning, because they told us the good and the bad. And at least me, I learned a lot, and I want to congratulate you. But I think that we need to understand more about this before you go to clinic.
Not much to add. I think the extreme phenotypes will give us a lot of molecular information. And in terms of general genetics for the cardiologist, it should really be used for increasing adherence to medication or changing medication. I think that's going to be the most useful part.
So Thank you all, and we'll take questions.